Good morning, everybody. My name is Tommy Boland. I'm the Associate Dean for Research, Innovation and Impact within the School of Agriculture and Food Science in University College Dublin. You're very welcome to this morning's research seminar, the fourth seminar in our series in the 2022 calendar year, entitled Infant Nutrition, Intakes and Impacts. I'm very excited about this morning's lineup, and we have two expert speakers to speak to you on this topic today. This morning's session will be chaired by Dr. Breege McNulty, an Assistant Professor in Human Nutrition in the School of Agriculture and Food Science. And I'll hand you over to Breege now.
Many thanks, Tommy. So it's a great pleasure to be here this morning and to be covering the topic infant nutrition intakes and impacts. It's really an important area, I suppose, of nutrition. I suppose we talk about nutrition through the life stages, but nutrition in infancy is really important for the proper development, both mentally and physically. And we know that it has important factors in determining risk later in adulthood. So it's really imperative that we research this area. So I'm delighted to be here this morning doing the seminar on this topic. So I'd like to firstly warmly welcome Dr. Alison Eldridge to give our first presentation of the morning. Dr. Eldridge has a PhD in nutritional sciences from the University of Arizona and completed her postdoc in nutritional epidemiology at the University of Minnesota. She currently is an expert scientist in the Nestlé Institute of Health Sciences in Lausanne in Switzerland, and also is an adjunct professor here in UCD. Her research emphasis at Nestlé Research is on dietary intakes, and she is principal investigator for the 9-country Feeding Infants and Toddlers study, and also the Kids Nutrition and Health study, which is sponsored by Nestlé. And today, Alison is going to be sharing her insights and challenges collecting dietary data on infants and young children. So I'd like to welcome Alison.
Thank you very much. Breege, thank you for the nice introduction. I am assuming that everybody can see me and my slides will soon appear. Very good, thank you. So we're here today to talk about infant nutrition intakes and impacts. Next slide, please. And I'll be speaking first to give you an overview of the Feeding Infants and Toddlers studies that Breege just mentioned. I'll talk about some key findings from our 9-country study, especially during this period of infant dietary transition, and then some challenges in dietary intake collection for infants with a focus on breast milk. That will transition us into Martina's presentation, which will really focus on lots of different aspects of breast milk. So between these two presentations, we're going to be going from a broad epidemiologic perspective into more of a cellular and specific perspective on the topic of breast milk consumption. Next slide, please. So what are the Feeding Infants and Toddlers and Kids Nutrition and Health studies? Well, basically, they're large-scale diet and behavior surveys that we've used at Nestlé to understand nutrient intakes and to help identify key nutrient gaps in the diets of children of all ages, from birth up to the age of about 13 years old. We can use the information from the studies to identify food intake patterns and potential feeding issues and gain insights on dietary intake and related behaviors. From our perspective, the scientific approach helps to build and share nutrition knowledge, and the sample sizes are generally large, which makes results generalizable. So far, since 2013, we've had 87 peer-reviewed publications from our studies in the 9 countries, and we've been able to foster science-based communication both with health professionals and consumers. But inside the company, we also use the findings to help provide a roadmap for product innovation and renovation for this important age group. Next slide, please. We've used a 3-tiered scientific approach for the Feeding Infants and Toddlers study. And by this we mean first understanding existing science in the countries where we've conducted this study, and then also, where possible, conducting additional analysis of existing national survey data. I want to underscore the importance of this for the field of nutrition, because many countries invest an enormous amount of resources in national surveys of dietary intakes and other health parameters, but they may lack some resources in terms of being able to publish all of the findings from the different national surveys. One answer to that question is what they've done with NHANES in the United States and have made the data sets basically publicly available. So any researchers who are interested in doing further analysis have access to the data. In Australia, you have access to the data if you're an academic institution, and this is also another way to get more out of the data sets. But for some countries, we've had to collaborate more directly with the ministries of health in the different countries to get access to the data sets, and then do our own analysis with an approach that's similar across all of our geographies. And where there are no national survey data, we've had to collect new data to fill gaps. And this has also given us an opportunity to look at some of the other aspects of infant feeding. So you can think of more diet and lifestyle attributes. Next slide, please. This is a map of the countries where we've done this study. We've studied almost 25,000 children from birth to the age of 48 months. And we've studied almost 55,000 kids from 4 to 13 years with a combination of national surveys, as I mentioned, and our own dietary intake data collection. Next slide. So what do we look at? Well, one of the aspects of the data collection is 24-hour recalls. And in all the 9 countries that we studied, we used at least one 24-hour recall for the dietary data collection. Most countries used 2 non-consecutive days, which gave us the ability to look at usual nutrient intakes. And the nutrient intakes were compared to estimated average requirements, or AIs, for vitamins and minerals. And then we can look at the percent who are consuming above recommended intakes for energy and other nutrients of concern like sodium. Because the 24-hour recall is pretty comprehensive, we're also able to look not only at nutrient intakes, but also food group intakes. So we can look at the percent of children who are consuming different food groups, how this changes by age, the particular food sources of energy and nutrients in the diet of infants and children, and the role of different product categories like milk or infant cereal in the diet. We can also look, this is maybe more appropriate for older children, but meal occasions. And we've done several studies on meal skipping patterns or eating and drinking between meals, so snacking habits. We've been able to look at portion sizes. And of course, in the countries where we've collected the dietary intake data, we also have a lifestyle and behavior questionnaire, so we can look at feeding practices that moms use when they're caring for young children, also physical activity and fitness and sleep patterns of older children. Next slide. So let's take a look at some of the findings for these different topic areas. The first one I'm going to focus on is nutrient intakes. And this example with a focus on iron compares findings that we've had from all of the FITS countries. You may have noticed from the map that we were able to collect data in 9 countries, but not all of them included infants. So this is just for infants 6 to 11 months old and 12 to 23 months old. And this shows a graph of the percent of children who are below the estimated average requirements for iron. You can see that this is higher in the 6 to 11 month olds, which is blue bars, but continues to be at a pretty high level in both the Philippines and Russia for the 12 to 23 month old period as well. And you might ask the question, why? One of the things that's important to know when you're looking at dietary intake data across countries is that you have to understand more about the local context and how things are actually calculated in the different countries. In Russia, for example, the estimated average requirement for iron is 10 milligrams per day for 7 to 12-month-olds, whereas in the United States, it's only 6.9. And in the US, it drops to 3 milligrams per day for 1 to 3-year-olds, but remains at 10 milligrams per day for Russia. And so because their standard is different for determining adequacy, they have a very high percentage of children who are inadequate using their standard, and that persists into toddlerhood. The issue is completely different for the Philippines. The Philippines has a diet that's based about 80% by energy on rice. And rice is not a great source of iron in the diet. And so these children transition from a breast milk diet, as you can imagine in infancy, into a primarily rice-based diet. And because they don't have very good iron sources in their diet at all, the level of inadequacy of iron intake continues to be high. So you have to understand the context of the country in order to interpret the findings. Next slide, please. So sticking with iron, but other nutrients as well, we can look at, well, what kind of foods might be able to help us reduce the iron inadequacy in the diet of children. And one of the things that's an important iron source in young children's diets is iron fortified infant cereal. And we can see in the little panel on the bottom, the percent of children who are below the estimated average requirement for iron if they consume cereal, very low, or if they don't consume infant cereal, and then it's about 70%. But the infant cereal consumers, and because infant cereal is fortified, also provides other nutrients. We can see that consistently across many nutrients in the diets of infants, they're higher in the cereal users. This is the percent of infants who are above the average intake level, and lower for the non-cereal users. Next slide, please. Another way to look at the diet is to look at the percent of energy that's consumed by different food groups. And what we can see from this graph is that as energy increases, we see a different pattern of consumption as kids go from birth up to the age of 48 months. We see in the blue bars, nearly all of the energy that's consumed for the young infants is milk products, which would include human milk, but the diet gets more diversified as time goes on. However, by about 2 years of age, the dietary patterns are pretty well established. Maybe even by 18 months of age, the dietary patterns are pretty well established. And we also see that the amounts of energy that are coming from meat, protein foods, grains, and sweets increases over time. Next slide, please. Because that previous slide showed that we're always changing the energy intake of infants as they grow, sometimes it's easier if you just look at the percent of total energy for major food groups. And we can see the dietary patterns established much more similarly here in this slide as the full milk diet switches to foods of the family table. Next slide, please. And here we see a similar graph, but this is for comparing different countries. So the top left is Russia, then we have China also on the top. The US is on the bottom left and Mexico on the bottom right. And we can see a couple of important differences here when we compare across countries. So yes, most of the infant diets are nearly fully milk. But there are some differences in the age categories. So for example, the US presents 0 to 6-month-olds, and whereas the first bar for Russia is 0 to 3-month-olds. And the first bar for China is 6 to 11-month-olds because the survey wasn't conducted in infants under 6 months in China. So it's important to keep in mind that we're not exactly comparing the first full blue bar at the same age categories. But then we can see a little differences in the patterns of diversification. So if you look at the red bar, for example, in Mexico, it represents, starting from 12 months, a pretty high percentage of the energy intake per day. And the red bar is actually showing sweets and snacks. And so we can see this is a problematic dietary pattern for children in this country, because even from a very young age, they're receiving quite a high percentage of sweets and snacks. Whereas if we look at the grain bar, so the yellow bar, we see a very high percentage of energy in China coming from grains, and also in the diets from Russia. Next slide, please. This slide shows 3 bars for each country, each of 5 countries, going from 6 to 11-month-olds to 12 to 23-month-olds, and then 24 to 47-month-olds for each of the countries. And what we can see from this one, in the blue part of the bars, is a variety of vegetables other than potatoes. And the orange part of the bars is the vegetable consumption that's related to potatoes only. So if you focus your eyes on the middle, where we're talking about Russia, you can see quite a high percentage of the vegetables that are consumed in Russia are from potatoes. Whereas if you compare to China, which is on the right-hand side, they eat a lot more different vegetables and actually relatively few potatoes. Okay, next slide, please. Sugar-sweetened beverages and sweets are introduced early, and we saw from a previous slide that this was especially the case in young children in Mexico, but also we see pretty high consumption of sweets by 24, so 2 years of age, and up in Russia as well. Next slide, please. And sodium intakes are often too high for many toddlers. So this graph shows 12 to 23-month-olds and 24 to 36-month-olds. And we can see 70% in the United States of these older children, the 2-year-olds, are consuming more sodium than required in the US and Mexico. A little bit less in Russia, and about 65% of children in China are consuming over the upper intake levels for sodium. Next slide, please. Finally, when we look at feeding patterns, we can see that there's early introduction of complementary foods. And this graph shows the percent of infants that are under 4 months of age that are receiving some kind of complementary food. We can see a lot of times it's grains. So in China, that would likely be rice, whereas in the United States, the grains that are introduced early are often infant cereals. Mothers in the US have a bad habit of adding infant cereal to bottles to help keep their children sleeping over through the nighttime. So we can see some early introduction of grains in that way from the US. And almost 6% of Mexican children consume sweets or sweetened beverages before 4 months of age. Next slide, please. Transitioning into our focus topic area for today, breastfeeding rates are generally lower across these countries than recommendations. Breastfeeding rates need to be improved in all countries to meet WHO recommendations for exclusive breastfeeding for the first 6 months. Next slide. So how do we measure breast milk intake in these large scale studies like the FITS study across these different countries? Well, in fact, direct estimates of breast milk intake are not feasible for these large studies. So we rely on estimates that have been derived both from weighing babies before and after intakes in smaller studies, and also in isotope methods that use deuterium to assess water balance between the mother and baby over a 14-day period. Next slide, please. And what we can see from some of the isotope studies, the picture on the left shows modeled human milk intake data from Dose the Mother deuterium studies by infant age group. And you can see they've split the infant ages into 1 month categories, 0 to 1 month, 1 to 2 months, and so on. And from these, estimates for birth to 5.9-month-olds and from 6 to 11.9-month-olds have been derived. So the FIT study that we've done in across the world has used these volumes as estimates for the total amount of breast milk consumed depending on the age or the feeding occasion when the children reach 1 year of age. Next, please. And so if no other sources of milk used, the values were assigned by age. But if they were partially breastfed, we actually subtract the amount that was fed in a bottle from the estimate of the 780 milliliters for the young babies and 600 milliliters per day for the older babies. But if the mom pumped breast milk and fed it from a bottle, we just used the volumes that were reported. So next slide, please. So finally, I'd just like to summarize, breastfeeding rates are low around the world. We saw inadequate iron intake in infants. We saw excessive consumption of sodium among toddlers, early introduction of some first complementary foods, and even early introduction of cow's milk in some countries. We saw that dietary patterns are set early and that consumption of sweets and sweetened beverages starts early in a lot of children around the world. There's inadequate vegetable consumption and also inadequate fruit consumption in many places. And few foods make up a significant portion of daily energy, such as rice in the Philippines. I've put my contact information here if you're interested in our list of publications, so please reach out. And finally, next slide. I'd like to say thank you to our many research collaborators, scientific advisors, team members, and participant families around the world to deliver on the FITS and kids studies. Thank you very much.
Thank you so much, Alison. That was a really, really interesting talk. So I just like to remind everyone, if you have any questions, please put them in the question and answer box and we'll come back to them at the very end. So I really welcome our next speaker, Dr. Martina Wallace, so my colleague, who's assistant professor in the Nutrition and Food Science section here in the School of Agriculture and Food Science in UCD. She completed her PhD with Professor Lorraine Brennan in UCD in the area of metabolomic-based biomarker discovery in assisted reproductive technologies. Then in 2013, she went to join the lab of Cristina Metalio at the University of California, San Diego, where her work focused on branched-chain amino acid metabolism in adipocytes and sphingolipid metabolism in macular telangiectasia type 2. Then in 2020, she came back to us here in UCD and she established her lab in the Conway Institute here. And currently, her research seeks to understand how altered metabolism contributes to adipose dysfunction and fibrosis and the impact of maternal obesity on breast milk composition and infant health. So I'm looking forward to hearing about your work, Martina.
No, sorry for the apologies for the delay there, just while I got the screen up. So thank you very much for the invitation to talk today and share what is somewhat preliminary data around our knowledge of fatty acid diversity in human milk, but an area that we're trying to do more studies in. So this is very much taking a different angle to Alison's presentation. So I'm mostly interested in, the nuts and bolts of metabolism, and this has brought me to trying to understand some of the metabolic pathways that support breast milk production. So the benefits of breastfeeding are very well established, as Alison just mentioned, and they're known, from the infant point of view. It reduces the occurrence of infectious diseases, can improve IQ, and can help protect from later life obesity. And it also has lots of benefits for the mother. So it decreases chances of breast cancer, ovarian cancer, can potentially decrease the chances of development of type 2 diabetes, and also improves birth spacing. And based on these benefits, it's actually previously been estimated that increasing breastfeeding may prevent as much as 823,000 child deaths and 20,000 breast cancer deaths per year. So it is very beneficial for both the infant and the mother. And key to some of these benefits is very specific components within the breast milk, which help support infant development, and also, the kind of hormonal and metabolic pathways that get activated within the mother to help produce breast milk. So when it comes to the composition of breast milk, our knowledge of it is constantly expanding. The key kind of chemical diversity, we have, breast milk is composed of a range of lipids, proteins, peptides, carbohydrates, and different micronutrients. And, they're well appreciated for the nutritional role, but also what's emerging more and more in the literature is key kind of bioactive and signaling roles of some of these components, from specific carbohydrates to specific lipids and peptides, that help kind of promote appropriate development of the infant. And what's also notable about breast milk is a lot of its components are distinctly enriched in breast milk versus other biological fluids. So, there's a lot of things in breast milk that aren't commonly, that you don't commonly come across when looking at other tissues and other biological fluids. And a good example of this is human milk oligosaccharides. So these are about the third most common component of human milk. And they've been shown to play a really important role as prebiotics and also as immunomodulatory components of the milk. And as technology is improving, and particularly around mass spectrometry technology, our knowledge of kind of the unique chemical diversity of human milk is also expanding. So in addition to kind of understanding what, there's a lot of interest in really properly profiling human milk and understanding its components, there's also a lot of interest in understanding how different maternal factors impact the composition of these beneficial components. So for example, how genetics, how maternal diet, and how maternal body composition might change levels of some of the beneficial components. And of particular interest is the impact of maternal obesity. So there is a rise worldwide in maternal obesity. And, one of the key questions is how does this impact lactation and milk composition? So there is some data indicating that it impairs lactogenesis. And maternal obesity has also been shown to be associated with increased risk for the infant or the child for later childhood obesity or metabolic syndrome as an adult. And, there's a lot of factors that play a role in this. So, the in utero environment and childhood environment, but there is also some data indicating from rodent studies where they do cross-fostering studies that particular components of the milk might also play an independent role and kind of have an impact on metabolic programming. So when it comes to milk, then I'm particularly interested in the fatty acid and the lipid composition. So human milk is composed of about 3 to 5% fat when you look at it as a weight per volume, but it accounts for about 50% of energy intake of young infants. And lipids are delivered in breast milk via this kind of unique structure called a milk fat globule. And this milk fat globule has this very distinct tri-layer membrane. So basically, as lipid droplets are made by the secretory cell, then as they exit the cell, they pick up the membrane from the cell. So they end up with a tri-layer membrane. And what this means is that these milk fat globules don't just contain this really important triacylglycerol rich core, which is really important for kind of the provision of energy-rich fatty acids, but it also means that it has this complex bilayer membrane full of different phospholipids and complex lipids that are known to play important signaling roles in other tissues. So, for example, in this paper here, which was published last year, the investigators looked at all the different species they could detect in human milk, and they had a really diverse range of trace glycerols, but also of a lot of other more polar lipids such as phospholipids, sphingomyelins, and gangliosides and ceramides. So all lipids that are known to play important signaling roles in other tissues, but really their role in human milk is less well appreciated. And then, so in addition to this kind of complex lipid diversity, the fatty acids within these lipids are also very diverse within human milk. And so the fatty acids in human milk primarily come from the diet or from de novo lipogenesis directly, or de novo fat synthesis directly in the mammary gland, or it can come from fatty acids that were made in the adipose or liver and then transported to the mammary gland. So when it comes to diet then, and considering how it impacts fatty acid diversity, the diet, can act as a direct source, for example, for essential fatty acids, which we can't make ourselves, just has to come from our diet and then be transferred to the milk in that way. Or our diet will also impact our own metabolic pathways and specifically impact our de novo fatty acid synthesis, which can then impact fatty acid diversity via impacts on our metabolism. And de novo fatty acid synthesis, it's primarily kind of being characterized in terms of the liver and our adipose tissue, but within the mammary gland as well, it plays a very important role. And the mammary gland actually has distinct adaptations for the change to type of fatty acids it makes. So in most tissues, our cells would use mammalian fatty acid synthase to make this straight chain palmitate, and palmitate is the primary product. Well, what's interesting about the mammary gland is it also contains the second protein called thioesterase 2. And basically, thioesterase 2 can interact with fatty acid synthase to hydrolyze the acyl chain when it's a shorter length. So it basically favors the production of more medium chain fatty acids, particularly in the range of about C8 to C14. So it modifies the product specificity of the fatty acid synthase. And this is important because mammary de novo lipogenesis is kind of uniquely adapted to make these. And this is evolutionary conserved. So you see this across most mammals where there's always a really high level of medium chain fatty acids within the milk. And it's important because the metabolic fate of medium chain fatty acids is generally different to that of longer chain fatty acids, such as palmitate. So they're much more easily oxidized, and they've also been shown to have some antimicrobial effects. So this is an important example of how mammary gland fatty acid synthesis is different. And I started working on, or started becoming interested in mammary gland de novo lipogenesis because my previous work was very much focused on de novo lipogenesis in adipose tissue. So one of our other primary lipogenic organs. And what we previously showed was that kind of in contrast to what's commonly taught in textbooks, where it's shown that fatty acid synthase will really only make palmitate, we found that fatty acid synthase is actually much more flexible than previously appreciated, and that when adipocytes upregulate the breakdown of branched amino acids, then they can use the intermediates of this pathway to initiate a new acyl chain. And then what this results in is that if you have increased accumulation of these intermediates, you end up with the synthesis of more of these monometal branched chain fatty acids, which are characterized by these, in this case, a distinct branch on the terminal end, or it can be on the penultimate end as well. And what this does in terms of the kind of chemical characteristics of the fatty acid is it makes them more fluidizing. So if you make more of these versus the amount of palmitate you make, it will contribute to more fluidity in the membranes. And it also means that they're resistant to oxidation. So most mammalian cells use monounsaturated or polyunsaturated fatty acids to increase fluidity in membranes, but these are sensitive to oxidation, whereas these branched chain fatty acids can provide an alternate method for this, which is not sensitive to that. And at the time when we were working on this, it was really presumed that all of the branched chain fatty acids that is typically detected in human plasma or tissue came solely from the diet because they are high in dairy products, but we were able to show that they're actively synthesized. And what was more important is we showed that the synthesis of these is decreased in an obese environment. So, you know, if we looked at these in obese mouse models as well as in humans, and we found that, you know, with obesity or with factors that play an important role in insulin resistance that comes with obesity, such as hypoxia and pro-inflammatory cytokines such as TNF-alpha, they all decrease the synthesis of these branched-chain fatty acids. And in general, it is now becoming more well appreciated that adipose de novo lipogenesis in of itself is very sensitive to obesity and a high-fat diet. So here I'm just showing some data where we quantify the amount of fatty acids that were made by de novo synthesis in high-fat diet versus low-fat diet-fed mice. And you can see that the high-fat diet completely suppresses de novo synthesis of fatty acids. So we're really interested, and we're continuing to work on, you know, the functional role of these branched-chain fatty acids, but we were interested whether this pathway plays an important role in breast milk fatty acid diversity as well, and if it might be impacted by maternal phenotype. So if an obese environment decreases DNL in the adipose tissue, does it also alter de novo lipogenesis in the mammary gland as well? And one of the other reasons that we were interested in breast milk from this point of view was that when it came to the branched-chain fatty acid literature, one of the main places where it had previously been measured was in human milk and different types of milk. So this has been kind of reported since about the 70s, and it's known to change with both geographical site with dietary intake. So dairy fat intake will change levels of this, and also with lactational stage. So they tend to be more enriched in the colostrum. And they're also really abundant in the vernix and the meconium. So the vernix is the waxy substance that covers the baby when it's born. So they seem to be really enriched in this kind of like early life stage. And there has been studies as well showing that they can, in rodent models of necrotizing enterocolitis, that they can reduce the incidence of this, and that they can also change the microbiota composition in this model, and also decrease the amount of inflammatory cytokines. So just having this branch on the fatty acid attached to different lipid groups can have these impacts. And then more recently, in the last 2 years, there's been a lot more data coming out now showing that if you have a branched-chain fatty acid on a lipid ligand, instead of a straight-chain fatty acid, such as palmitate, that it can be immunomodulatory and it can change the behavior of dendritic cells and also NKT cells. So based on this data, you know, we're really interested to see, first of all, are branched-chain fatty acids de novo made beneficial components of breast milk? So are the branched-chain fatty acids in breast milk primarily diet-derived, or can de novo synthesis also contribute to their levels? And how does maternal phenotype impact mammary de novo lipogenesis? So to start to look at this, we collaborated with Africa Sullivan here in UCD, and I started this when I was a postdoc in UCSD. And what Africa had done was collected breast milk from a cohort of women, where she tracked 48 women throughout pregnancy, and then collected samples from 36 women, 1 month postpartum, who breastfed, as well as another 12 women who formula fed. So they just provided the formula that they fed to their babies. And so Africa gave these samples to us then, and what we did is we performed an untargeted GCMS analysis, which was very much optimized for isomer separation. So we used a specific column that allowed us to try and separate out the fatty acids as much as possible, so we could really capture as much of the fatty acid diversity as possible. And we compared these against a mother's gestational weight gain recommendation, so whether they were within or above what was recommended, and their trimester 1 BMI and infant anthropometrics. And so the first thing we noticed is that, you know, that the diversity of fatty acids in this milk from this Irish cohort was very diverse, similar to what has been reported in other papers. So we detected 44 different species. And this is comparison to, you know, typically if you look at plasma or a lot of other tissues, the max number of species you're going to see is about 34. So there is a lot more diversity in the milk. And when we look at the representation across the main different fatty acid groups, you see that the MUFAs, or monounsaturated fatty acids, have the most species with 12. Then we detected about 9 different long-chain fatty acids, with palmitate being the most abundant. And we detected 7 different branched-chain fatty acids as well. So this is more branched-chain fatty acids than what we had seen previously in adipose tissue. And they tended to be more of that medium chain length, similar to what is kind of previously been shown with straight-chain fatty acids. And when you look at the molar percent then, so the actual amount of the different classes in the milk, we found that about 57% was represented by the monounsaturated fatty acids and about 28% by long chain fatty acids. And the two most abundant fatty acids were oleic acid and palmitate. So overall then, monounsaturated fatty acids seem to be the most abundant and have much greater kind of species diversity. And what's important to note is that, you don't normally see the species diversity in plasma. So it's really, substantial kind of mammary gland metabolism and like desaturase activity that contributes to this increased monounsaturated fatty acid kind of diversity within the milk. So then we focused in on the branched chain fatty acids, and although they only represent as a molar percent about 0.6%, because milk is so fatty acid rich, they're actually present at millimolar levels within the milk. So there's a wide range in levels across the different samples, and we also then compared levels to infant formula, and the infant formula we collected represented about 6 different brands. And you can see that the branched chain fatty acids were nearly completely undetectable in the infant formula. And this is in contrast to a lot of other fatty acids where they're actually quite well matched. So the more common fatty acids, such as the medium chain fatty acids C12 and C14, along with palmitate, the average in breast milk versus the infant formula was quite similar. So then finally, we wanted to look at how levels of the fatty acids change with different phenotypic factors. And just to note, in terms of the demographics of this group of women, they all started out with very healthy BMIs. None of them were overweight, and they had an average BMI of about 22. And what Africa and her students decided to do was separate these out based on recommended gestational weight gain. So they looked at those who gained below the recommendation, those who gained within, and those who gained above. And then we compared the fatty acid profile across those groups. And what we found is that the biggest difference was when we compared those with a high versus a low gestational weight gain. And the main fatty acids that were different between these two groups were the branched chain fatty acids. So here you can see the 7 different species clustering together in this volcano plot. So branched chain fatty acids were the most decreased fatty acids in women who gained above the recommended gestational weight. So this is kind of, interesting initial data indicating that the levels of branched chain fatty acids can change with maternal phenotypic factors. And we're looking to try and see if we can expand this out and look at larger cohorts with more maternal obesity. But in the meantime, we decided to go back to our experimental mouse models and see whether or not we could see or prove if these can be de novo made by the mammary gland in these models. So we asked the question, does de novo lipogenesis contribute to BCFA levels in milk, and is it decreased by excess gestational weight gain? And so in order to do this, we designed this study where we used black 6 mice, commonly used in these diet-induced obese models. And we gave them, after conception, we gave them either a high-fat diet or a low-fat diet. And then we continued this diet through 10 days kind of post-parturition when they had their pups. And what we did was 3 days before we collected the milk from the dams, we injected the dams with heavy labeled water, D2O, as Alison had mentioned earlier, is a really important method for looking at the transfer or the consumption of milk by the babies. And in this case, we could use it to look at milk consumption by the pups. But in addition to that, we can also use the heavy labeled water to calculate the amount of de novo made fatty acids in the milk. And so what we found when we looked at these mice afterwards is that, the dietary intervention did increase dam weight. And this is the weight of the dams the day after they had given birth. And then when we looked at, by looking at the transfer of D2O from the dam to the pups and using that to estimate milk consumption, we could see that the pups on the high-fat diet consumed less milk. And we also saw a trend towards decreased growth in these pups. And this is kind of in line with what has previously been reported in humans as well, where there may be impaired lactogenesis with obesity. So then when it came to looking at the amount of de novo made fatty acids in the milk, so using heavy labeled water, you can calculate the amount of de novo made fatty acids because the heavy labeled water gets incorporated into the acyl chain as it's being made. So you're able to use some kind of straightforward calculations to say how much has been de novo made. When we looked at branched chain fatty acids, we saw significant synthesis of the branched chain fatty acids in the milk. And this is important again, because previously, it has been primarily presumed that most branched chain fatty acids come from the diet. But here we can see that, there is a significant amount of the de novo made fatty acids in the milk. And also what's important is this method doesn't discriminate if potentially they were made in the adipose tissue and transported, but because we're seeing these kind of shorter or medium chain branched chain fatty acids as well, this is more likely to come from the mammary gland. So then we looked across all the different fatty acids that could be de novo made. And here in white, I'm showing their synthesis on the low-fat diet and in gray on the high-fat diet. So all of these values are normalized to the value of the low-fat diet. And you can see that there's a general suppression of all fatty acid synthesis. So similar to adipose tissue, this high-fat diet seems to be suppressing de novo lipogenesis. And this is in contrast to what's typically found in the liver, one of the other main lipogenic organs, where a high-fat diet can increase de novo lipogenesis. And then the last question was, well, how does this change in de novo lipogenesis impact overall fatty acid composition of the milk? So in this graph here, I've plotted each of the fatty acids based on their increasing chain length. And then I've normalized all the fatty acids to the levels in the low-fat diet, which is the dotted line here. And what you can see is this general trend where there's decreased levels of medium chain fatty acids and some long chain fatty acids up to about C16, but then there's increased levels of all the longer chain fatty acids and very long chain fatty acids. So those that are more likely to come from the diet. So in these high fat diets, the main fat composition fat source tends to be lard, and these tends to be much more enriched in these longer chain fatty acids. So you can see that shifting the fatty acid composition or increasing the fat composition, decreases the amount of fatty acids that typically come from de novo synthesis, and instead you get a lot more that's coming directly from the diet. So then finally, just as conclusions, so this is our initial kind of data trying to understand the potential role of these branched chain fatty acids in human milk. But I think overall, as a conclusion, de novo lipogenesis and desaturase activity in mammary glands is a really important contributor to fatty acid diversity observed in human milk. And mammary DNL and the resultant fatty acids may be sensitive to maternal obesity or inhibited by it, similar to adipose DNL. So this is kind of becoming more well established in adipose tissue, where an obese environment decreases DNL, but I think there really hasn't been as much data looking at this to see if obesity or maternal obesity impacts mammary DNL in a similar way. And then finally, when it comes to branched chain fatty acids, it's well known now that these are components of the milk fatty acid composition. And there has been previous work showing the really important contribution of diet to branched chain fatty acids in the milk, and specifically of different dairy products. But I think, our data indicates that potentially these can also be de novo synthesized, and then therefore alterations in de novo lipogenesis will also impact levels of branched chain fatty acids in the milk. Then when it comes to function, there is emerging data indicating they might be immunomodulatory. They might play a role in metabolic programming. So in data I haven't shown today, we know that they can also act as PPAR alpha agonists. And then finally, that they might play a role in microbiota modulation. And as more groups continue to look at these branched chain fatty acids, I'm sure we'll start to see the more evidence of the role they play and why they may be important for infant health. And then finally, for future directions, we really have to look at levels of these fatty acids and of de novo made fatty acids in general, in larger cohorts with maternal obesity or excess gestational weight gain, or also with gestational diabetes. And as Alison mentioned earlier, D2O is commonly used or is used in women to look at babies' milk intake, but these methods can also be used to quantify the amount of de novo made fatty acids in the milk. And then finally, much more mechanistic studies are needed to determine how altering levels of branched chain fatty acids and other DNL-derived fatty acids impact immune development and metabolic programming. And then as a final note, there is a number of challenges associated with measuring these BCFAs, or reasons why they may have been overlooked in the literature in the past. So these are generally isomers of much more abundant fatty acids, so they can be easily overlooked, and most LC-MS lipidomic platforms lack the resolution to identify these BCFAs. But this is changing now, especially as technology is improving, and you see more and more studies started to report these. So then finally, the most important slides to acknowledge our collaborators who worked with this. So Africa Sullivan and the different students in her lab who collected and worked on the human milk study. And then I carried out the animal studies while I was still in Christian Metallo's lab in UCSD, and there is a number of undergraduates there who helped me with these measurements, specifically Noah, Jivani, and Michelle. And then finally, our initial BCFA work was funded by Ajinomoto, and then the animal studies looking at milk de novo synthesis was funded by seed funding from the UCSD Mother's Milk Center of Excellence. And with that, I'd be happy to take any questions. And again, thank you very much for your time today.
Thank you so much, Martina. That was a really, really interesting presentation. Just to reiterate, if you do have any questions, if you want to pop them into the question and answer box, just at the bottom of your screen. But just to start us off, I think I'm just going to ask, because I found it really interesting, Alison, in your presentation, I suppose, having that broad overview on a global basis of all of those different countries. And I was just wondering, in terms of the iron, because I know you focused in at the start, is it that cultural, because I know in Ireland here, it's the lack of, or sometimes we don't introduce meat because there is this kind of thinking in mums that potentially they don't, or, it's a bad thing to introduce it too early, but we know the iron stores kind of go at 6 months. Is it that you see potentially in different countries as well?
I think that there is a real opportunity for education about introducing meat early, but as you said, not very many people do it. And I have to say, the baby products for meat are not recognized as being culinarily, let's say, very interesting. So we have to get over the mom's hesitation on introducing actual meats from the family table prepared in a way that is digestible for babies.
Yeah. And Martina, there's a question here for you. In how important is dairy intake compared with de novo synthesis of branched chain fatty acids production in breast milk, do you think?
So really, we just don't have the data to answer that question. So dairy intake is definitely important for BCFA levels. And then also the endogenous metabolism of the BCFAs that come from the diet. So previously we found that they tend to be used up by the liver quite quickly and maybe don't make it all the way to the mammary gland. But again, I think they just haven't been explored enough. So the relative contribution and whether one is more important than the other is not known, but it's likely that both are important contributing factors.
And Alison, your data showed that China has much lower levels of consumption of sugar-sweetened beverages than Mexico, for example. Did this translate into differences in obesity rates when you were looking at it?
Yeah, that's a really good question. I can't say that our data actually generated. We didn't always have the ability to evaluate obesity rates ourselves with our data collection. So if it was just questionnaire and 24-hour recalls, for example, we didn't necessarily measure. But I am aware of other published studies that have indicated that the obesity rates are higher in Mexico than China. So it follows definitely.
And just onto that, I was interested in the patterns, the dietary patterns that they change over time. Did you look in terms of whether that relates to adult dietary patterns in that particular country? Like, are they reflective at that age of the actual adult patterns that we would see?
That sounds like a really good follow-up research topic. Thank you.
Firstly, and just a question for you, Martina. Congratulations on your presentation. Could you speak more about the immodulation effect that you think the BCFAs might have?
Yeah, so the potential kind of immunomodulatory effect is really kind of an emerging area. So there's not a lot of data out on it yet. What is kind of coming out as most prominent is so branched-chain fatty acids are most well known as actually components of bacterial membranes. And here's where this kind of strongest data is coming out, that if they're present as part of certain lipids, that they modulate immune function differently to if there's a straight chain. Now, and when it comes from the interim point of view, the strongest data has been in that paper I cited on necrotizing enterocolitis. So again, it's an emerging area, and it's been slowed down because of they're not always typically measured, and also it's really hard to get standards of these branched-chain fatty acids, so you can do the interventions that are needed to be done to proper mechanistically connect these. So far it seems promising, but lots to be done.
Yeah. And just another one for you, Martina, in terms of collecting the human milk samples, is there a particular way that they need to be collected, stored, in order for you to account for the fatty acid analysis later on?
Yeah, so it really depends on which fatty acids. So those like polyunsaturated fatty acids, which are more easily oxidized, will be much more sensitive to storage and collection. In terms of branched-chain fatty acids and saturated fatty acids, they tend to be more stable because they don't have those double bonds that can get oxidized. So again, it's very kind of lipid species and fatty acid specific, but in general, for the BCFAs, we find they're quite stable.
Okay. And just another question for you, Alison. How much does differences in stable foods between countries account for variations in the interpretation of the data?
Yeah, that's a really good question. I would say having an understanding of the cultural perspective of the foods that are available and how they're consumed by infants and children is hugely important for interpreting the data. For example, one of the things that we're often faced with low intakes of dietary fiber. But we don't see that in Mexico because so much of the diet in Mexico is based on corn masa, and corn masa actually has quite a lot of dietary fiber. And so, our people say, oh, well, all children need to eat more fiber, or all, they try and make these big generalizations, and no, they're really country specific. If 80% of the diet is coming from rice in the Philippines, we have huge challenges to feed those children. And they're not just lacking meat in their diet, they're lacking fruits and vegetables in their diet, even though they live close to the equator and fruits and vegetables can easily grow in their climate. But they're just not incorporated at high enough levels. So I think it's really, it's a really important point. And like I said, with Russia, we have to understand the context, not only the context of the foods that are available, but also what are the nutrient recommendations in the different countries in order to interpret the findings.
And just out of that, Alison, did you look at it in terms of having one recommendation, or what was the decision to use the country-specific recommendations in your report?
We did try and do that, and actually for many countries that don't have recommendations, we applied the recommendations from IOM. They're already used quite widely in Central and South America, and the Philippines have adopted recommendations that are similar to IOM recommendations. So we did apply those. But when a country had their own recommendations, for a paper to be relevant for the nutritionists in their country, we had to actually use their recommendations in publications.
Yeah. Thank you. And Martina, there's just a query to clarify whether LCFAs are increased with an obesogenic environment as well as reducing BCFAs.
Yeah, so on this, in our mouse studies, the main long-chain fatty acid C16. So this actually wasn't increased in the milk of the high-fat diet-fed mice, but the longer chain fatty acids, once you get up to C18, that's when you really start to see it, and that reflects what's in the composition of the diet. In terms of human studies, so because we look primarily at gestational weight gain, we didn't have, you know, a study that we could address obesity, maternal obesity with. But looking across the literature, there's really no really strong consensus of changes in the fatty acid diversity so far. There's not enough studies yet, probably. And also, one of the complications with this kind of analysis across studies is that, you know, fatty acids are typically presented as a percentage of the total fatty acids detected. And because different people detect different numbers of fatty acids, it's kind of creates more problems with comparing across studies. But what I have seen from the literature is that the medium chain fatty acids do seem to be some evidence showing they're decreased in the milk from associated maternal obesity, but I haven't seen as much of a strong, or I'm not sure about the longer chain fatty acids.
And just one last question, because it is the last question. In terms of collecting sample, again, Martina, for you, do you need to have a full breast expression of aliquot so that you can calculate the total fat composition, or will it be sufficient to have just a sample from the start of the feed, etc.? Because I know it varies depending then.
Yeah, I think this is you know, there's a lot of opinions probably on this. In the study we did with Afric, they were, the milk was, because it's very well known, right, that at the start of the feed, it has a different milk, a total milk amount versus the end of the feed. And that's why people normalize fatty acids the way they do as a percent of total, because the amount of fat can vary so much depending on when the last feed was, where you are in the feed, and so on. Mm-hmm. So in the study with Afric, what they did is they specified for the mother to kind of feed normally for 2 minutes and then stop briefly and then collect 15 mils of milk and then continue. So it's kind of like a mid-feed sample. So really, you know, there's a lot of different ways you can look at it. And the idea behind normalizing as a percent of total is to avoid some of those big shifts in the total amount of fat that can occur due to, you know, variation throughout the day. So I don't think there's a straightforward answer to this, unless we do like a I think this is kind of like a much broader topic.
Yeah. That's perfect. Well, just finally, I want to thank both Alison and Martina for wonderful presentations, really interesting. It was really informative session. And also to thank Professor Tommy Boland, who organises, and Patricia behind the scenes, who've done a lot of the organisation, and obviously the committee that's involved in it. So thank you very much.
Thank you for the invitation. It was a pleasure.
And also just to note before we go that this session was recorded. So all attendees will be sent the link to the recording, and it will be made available on the Schools of Agriculture and Food Sciences website. So I think we'll leave it there for today.